Good afternoon, everybody. Michael Crawford from the Soil CRC, where I'm the CEO. My role here is briefly just to welcome you all, to introduce you to the webinar, and say a couple of words of introduction. In doing so, I'd firstly like to acknowledge traditional owners of the lands upon which we meet, wherever we are in Australia, and I pay my respects to elders past and present. Before jumping into the webinar itself, just a couple of words around the Soil CRC. Looking at the registrations, we have a very broad, diverse range of participants in this webinar of registrations, and many of whom have been engaged with the Soil CRC at different times, but many of you might not know too much about the Soil CRC. So the Soil CRC essentially is a collaboration of farmers, industry, and scientists involving 39 partners across Australia, 8 universities, 4 government agencies, 7 industry partners, and 20 grower groups. We're funded by the Australian government and by our partners to deliver research that helps farmers improve their soil performance and increase their productivity and profitability. And today's webinar will be an example of some of the work that we have been doing. Essentially, we've been funded for 10 years. We commenced back in 2017, and that comes to an end in June next year, 2027. So we're coming into the last year of the CRC with a large focus now on communicating, disseminating the research that has been undertaken through the Soil CRC. Collectively over that period, we have attracted and have invested $70 million of cash, and that's been matched by $120 million of in-kind from our various participants, making it the largest collaborative soil research effort in Australia's history. The participants in the CRC, as I said, we have our major partners, our associates, 8 universities. You can see the logos there, a few state government agencies, some other industry partners, and importantly, those 20 grower groups from all states of Australia. So if you have a close look, there'll be many of the logos there that you see represented on that slide. It gives you an idea of who's actually been a part of this collaborative effort over the last 9 years and over the next year. That's the background and context. I'll just say a few words about housekeeping. The way we're operating this webinar, it's been recorded. It will be available at the Soil CRC Knowledge Hub for those to look at afterwards, along with, dare I say, a whole plethora of other resources that might attract your interest. During the webinar, your cameras as participants will remain off and your microphones muted. If you have questions, I really do encourage you to ask questions. Type them into the Q&A box. We also have a chat box. Use that to introduce yourself, tell us where you're joining from, sharing other relevant insights. But don't put the questions there, put the questions in the Q&A box. We will run this for 60 minutes, including the Q&A and discussion. And after the webinar, there'll be a short survey to make the next session better for you, because we do value your insights. So I thank you for participation and encourage you to enjoy this webinar. As I introduce the speakers, on the topic of unlocking the potential of sandy soils, we have Professor Richard Bell from Murdoch University, a very esteemed soil scientist over the past 40 years now, an Emeritus Professor there at Murdoch University, who's been leading some of our research. We have Dr Amanda Schapel from Prime Industries Resources South Australia, again, a well-qualified soil scientist and who's been working on some of our projects and others. Then Stephen Lang and Tania Monir, who have both been PhD students in the Soil CRC, and our PhD program is a big feature of it. Stephen's at Adelaide University. Tania has qualified now and Dr Tania Monir from Murdoch University. They'll both talk to some of their research as well. The research we're talking about draws largely upon Soil CRC work, but also will touch upon other activities funded by GRDC, by West Australian Department of Prime Minister's Regional Development, by Future Drought Fund. But largely it's about the Soil CRC activities. So in saying that, I'll now pass across to Richard.
Thank you, Michael. Why are we interested in unlocking the potential of sands? The key takeaway here is that sands have low reactive surface area, so that's the key thing I want you to recall. And that underpins most of the issues that we end up dealing with in sands, whether it's low water retention, low nutrient availability, low organic matter, water repellents, poor pH buffering, or compaction. But not all sands are the same. We can classify sands as having anything from less than 1% up to 10% clay. And within that range, we get a huge variation in behavior of sands. A sand with 1% clay can behave very differently even from a sand with 3%. There are differences in sand grain size, in the coatings on sand grains, in the pH, and rainfall and drainage of the site can also influence sands, particularly by affecting organic matter. But they all have low reactive surface area. Here's 3 examples of sand profiles. The one in the centre with less than 3%, and the one on the left, 3 to 6%, and the one on the right, 6 to 8%. But they all have low cation exchange capacity, which is a good proxy or indicator of low reactive surface area. Low plant available water content, though, is one of the major drivers of differences and the behaviour of sands compared to other soils. This table here shows the plant available water content in the top metre of the soil profile. So you can see a medium grain size sand will store 40 to 50 millimetres of water per millimetre depth, whereas a loamy sand, 60 to 90 millimetres, and a sandy loam, 60 to 100 millimetres. So these differences can have quite profound influences on crop productivity. But rooting depth also matters. Even relatively small decreases in the depth to which roots are able to penetrate decreases that plant available water content. There are many types of amendments that can be applied to sands, and in today's presentation we'll deal with quite a few of those. 5 main groups, but within each of those groups there are different types. The next question is, where are the sands? Nathan Robinson and colleagues across southern Australian states did a remapping of sands, and they came up with 16 million hectares in agricultural land across southern Australia. As you might expect, most of that's in Western Australia, and 51% of the agricultural land in the state are indeed sandy soils, but large areas also in South Australia and significant areas in New South Wales, Victoria, and Tasmania. So at this point, I'm going to hand over to Amanda to talk about some of the South Australian findings that she has to report.
Thanks, Richard. So I'll get to talk to you about the results of a meta-analysis that we did. And the meta-analysis is just a fancy way of saying that we collated a range of trial and demonstration sites from across South Australia, Western Australia, and Victoria that had results for sandy soils that had applied either subsoil clay or organic amendments such as manures, compost, and biochar. Now, I have a note that biochar, we've only got a couple of those samples, so we don't have too many. But what we did with that, we wanted to try and query the data to find some take-homes that we can have some practical rule of thumbs for people to take along with them. One of the key things that we found was that the depth of sand really does matter. So a lot of the production information that we have here is from broadacre cropping, but we do have some pasture sites as well, and we use biomass as our measure of productivity on those instances. The graph that we can see there shows the differences in yield, which is going up on the y-axis there. And down the bottom, we have the depth of sand over clay. So we have a shallow sand with less than 30 centimetres. Then we have different classes from 30 to 60, 60 to 90, and greater than 90 centimetres before we hit clay. The little dots there show you what the average tonnage is, and the numbers tell you how many records that we had. So we had 89 projects and we had 270 records, so a number of treatments and a number of different times for these ones. The grey line there is showing that as these sands get deeper, we have a loss of productivity. And we know that as we hit sand hills, we get less yield on those. What was really interesting to note was that green line that shows there is what happens when you applied organic amendments like compost or manures, is that you can maintain that productivity on some of these deeper sands, particularly those ones greater than 90 centimetres, so that production can still stay the same. When we looked at subsoil clay addition to sites, we found that the production really only improved where we had subsoil clay between 30 and 60 centimetres from the surface. So that was where we got the best results. When we talk about organic carbon, it was really interesting to see that when we had organic carbon, Walkley Black, your standard soil test result, in the top 10 centimetres across all of those sand classes, you didn't get a real change or no statistical difference, at least in carbon values across all of those sites in that top 10 centimetres. So they were all fairly similar. But it was when we looked deeper than that, so in that 10 to 30 centimetres, that we did notice there was quite a big loss when you have clay deeper than 60 centimetres in the profile, so those deep sands. You can have a significant loss of up to 0.5% of carbon. So that really comes down to the inputs that you would get within those deeper sands. And probably a thing that we note here that when we do sandy soils and ameliorations, you often, if you only measure the top 10 centimetres, you miss a lot of the story. So it's really handy to try and measure that little bit deeper, 10 to 20 or 20 to 30, or you can do a 10 to 30 centimetre sample. It really does provide a lot of context for when we're measuring changes, particularly in carbon. The other thing that came out pretty clearly is the longevity of effect. So productivity shows up really quickly, and we know that when we see trials, we often get a result in the first or second year. They stabilise often by 5 years. When we were adding organics, organic amendments, so this was often compost and manures without subsoil clay, the productivity was maintained for a shorter period. So at 3, maybe 4 years, you had a response, but then it declined. And this comes to be about sands and that ability of them to protect or stabilise that carbon in that soil. When we looked at subsoil clay addition, we saw that carbon gains build really slowly. So we could see really sharp increases in productivity pretty quickly, and we measure those in these short-term projects that we often have. But when we look at subsoil clay, we need to look longer. So it's at least 5 and often 10 years, particularly if in a low rainfall environment. Some of the great work conducted by Western Australia by David Hall and Stephen Davies has shown that we can have those carbon changes stabilised for over 15 years. An interesting finding that came up out of the data, so we had lots of different information, and one of them was iron. So it consistently found that products that had higher iron often had higher productivity and carbon compared to their control treatment. So this information was then transferred into the Western Australian trial that Richard will talk about a little bit later. We always hear the story that if you improve soil carbon, you can improve productivity, and we really wanted to test this in sandy soils. One of the first things that we looked at is what are the key drivers of productivity, and no brainer, it was rainfall. So winter and summer were really important for both. What was interesting was the biggest change in carbon actually happened to be summer rainfall, which led to the biggest loss of carbon. And this is generally because most of our southern areas have temperate conditions over summer, so hot and dry, so you don't get a lot of turnover of carbon. The microbial activity is very low. If we get a wet rainfall or moisture coming into the system and we have warm soils, we actually get a lot more decomposition and often loss to the system, again, because our sandy soils can't protect or stabilise that carbon. What we did find though is that there is a bit of a sweet spot in the top 10 centimetres where we can see a change in carbon, and that happens to be between 0.75 and 1.5% carbon of your Walkley Black test. So what this means is that we kind of have a bit of a threshold. So below 0.75% in the top 10 centimetres, our soils aren't functioning. They're not being able to provide those ecosystem services such as nutrient availability in the situation. So they are basically starve the biology. So if we wanted to improve that, we would select amendments that would help to rebuild that function. So we'd need to get that system kick-started. If your carbon values are often above 1.5%, there is enough in there that's adequate for soil function already. So what we often find is that improving the carbon wasn't the key constraining factor for changes in productivity. It was production constraints. These could be things like pH, salinity. So in those instances, you'd be looking for amendments that really address those specific constraints. If your carbon results do happen to fall in between that 0.75 and 1.5, and particularly if you're at that lower end, there is an opportunity to improve carbon, to improve your productivity with a lift in carbon. And in these situations, we would be looking or choosing an amendment that's based on 1, practicality, 2, on cost, and 3, on what the other constraint can be. And in South Australian soils, we're often finding that acidification or acidity is probably one of our key constraints for limiting in this area. So when we looked at subsoil clay, there were some really clear-cut cases. Productivity is clearly linked with an increase in carbon or clay concentration up to 15%. As soon as we hit 15%, there was a really rapid drop. So if you're ever asked if can you apply too much clay? The answer is definitely yes. Once you get above 15%, you're hitting those values. Unfortunately, this is when we actually started to see organic carbon increases in the soil. So there's that flip side about when we're talking about adding subsoil clay, about having that balance of still having enough productivity. Again, when we're looking at organic carbon, the biggest increases didn't often occur in that top 10 centimetres. They were in that 10 to 30 centimetres. So when we talk clay sweet spots, or when we want to target it, is between 6 and 15, or probably 6 and 12%. And you'd fine tune it for rainfall. So definitely on the lower end if you have low rainfall. And what you're trying to do there is overcome the constraint without tying up that water. If you're going up to that higher 12 to 15%, I would recommend that in higher rainfall environments where you have plenty of organic matter inputs in the systems. And this often suits some of those higher pasture biomass increases in those ends. And this now segues back into Richard's results on talking about subsoil clay and the trials.
Thanks, Amanda. That's really a good background to help us understand what we've learned from past research on adding clay and adding organic matter. What I'm going to deal with now is some new experiments that we've done, principally with Soil CRC funding, and our main partners in that work are shown in the slide. Adding subsoil clay is a proven way of improving unlocking the potential of sands. The questions are, is that clay available on site? And to be useful, it needs to be within 2 to 5 meters depth. If it's too deep, it's too expensive to get it out. It also needs to be within close range, preferably within a couple of 100 meters of where you want to spread it. And ideally, it would be upslope of where you want to spread it so that you're carting clay downhill rather than uphill. So there are 2 main approaches. We can either dig, cart, spread and incorporate, or delve and incorporate. to increase the clay content in the upper horizon. And the photograph on the left shows the benefit for crop establishment with adding subsoil clay here by reducing water repellents. This is a site at Esperance, and the other photographs just show the typical soil on which that clay amendment is happening and the machinery that was used to dig the pits. David Hall's experiment at Esperance that went for over 15 years really shows the benefits of the subsoil clay addition. This is 15 years average responses where subsoil clay at 200 to 300 tonnes per hectare is producing about a 50% yield increase averaged over that time, which is very impressive. Only 50 tonnes of subsoil clay didn't have any significant benefit. But interestingly, even the best yields there are still below the water limited yield potential for that particular location. It's important, though, to understand what are the properties of the subsoil clay before it's used. David did a survey of pits that have been used for claying, mostly in the southern region of the West Australian wheat belt, 82 sites. The mean properties look pretty good, but if you look at the minimum values across that survey, some contain next to no clay, low pH, very little potassium or sulfur, low cation exchange, or low phosphate retention. So unless you are confident you've got a good type of clay in the subsoil, it's an expensive operation, and you should look very closely at whether it's going to pay off. And a simple way in which you can check whether the clay content is going to be suitable is the ribbon test. And if you haven't done the ribbon test or need some refreshers on how to do the ribbon test, you can go to the Soil Quality Knowledge Base, which is on the Soils West website, and that will give you some methodology on how you can, in the field, quickly assess whether the clay content is going to be enough. So if subsoil clay works, why would we look at other amendments? Well, subsoil clay is not always available. It is quite an expensive operation to add clay. And we were interested in the question of whether there are high activity amendments that you could apply at much lower rates and still get the same benefit. And there are some amendments that would address constraints that are not treated by subsoil. So what I'm going to do is go through a number of these novel amendments that we have experimented with in our Soil CRC projects. First, bentonite. It's a high activity clay, and the clay that's mined is pretty undiluted by anything else. It's fairly pure clay, generally alkaline with good contents of potassium, sulfur. But note there's also quite high exchangeable sodium and boron, and we have found in pot experiments that plant uptake of sodium and boron at 40 tonnes per hectare addition does result in a reasonably high uptake into the plant, so you wouldn't want to over-apply that amendment. So far, in our field experiment, we didn't get any yield response to a 15 tonne per hectare addition. Hydrotalcite is another product. It's an interesting clay-type material because it's positively charged, unlike clay, most clays, which are negatively charged. So it has the potential to adsorb nitrate, sulfate, and phosphate. And in our pot experiments, we certainly have been able to show that it increases retention of phosphate, but can decrease plant phosphate uptake. In our field experiments, 10 tonne per hectare addition. After 4 years of application, we haven't seen any yield response there. Ironman gypsum is a byproduct of synthetic rutile production. It's basically gypsum, but has a high content of iron and manganese oxides. So here we were interested in the iron oxide content, following up the meta-analysis results that Amanda showed you, so that we have the potential to sequester carbon and limit phosphate leaching. 2 field experiments we've done. So far we've seen no yield responses, but quite a big increase in sulfur uptake with the Ironman gypsum. Zeolite is a aluminosilicate rock mineral with a high surface area and cation exchange capacity. Our pot experiments show that at 20 tonnes per hectare, we do get increased water retention and decreased leaching of N, P and K. 1 field experiment at Wotherngara, 5 years after application, we have a yield increase there at 15 tonnes per hectare application. But at another of our sites at Quetta, 10 to 30 tonnes per hectare. After 4 years, we've seen no yield increases there. Next, biochar. There's been a lot of hype about biochar and an avalanche of publications on it in the last 2 to 3 decades. But just note, there are many types of biochar depending on the feedstock used. So we can't say what biochar did unless we're specific about the type of biochar, just as it doesn't make any sense to say fertilizer did this or fertilizer did that without being specific about the type of fertilizer. So this experiment done at Esperance on the sand, we had wheat straw biochar and chicken manure biochars, both at 10 tonnes per hectare. In the first year, we got significant increases in yield of wheat. In the second year, we're not getting any increase with the wheat straw biochar, but the chicken manure biochar continues to increase the yield. In the third year, those yield responses had disappeared. But interestingly, in the same experiment, phosphate fertilizer responses continued in every single year. So what we concluded was that those biochars are both reasonably nutrient rich, but by the end of 2 years, most of the readily available nutrients had been exhausted, and so the other benefits of biochar weren't being expressed once the nutrient benefit no longer was occurring. I'd like to share a couple of case studies of other experiments that we've done. Firstly, at Quetta, which is in our central wheat belt. This was an experiment, an on-farm scale experiment with big plots using farm machinery, where we've compared on a deep sandy duplex soil the bednar treatment, which is basically a deep ripping and topsoil mixing operation in one, and then adding compost or gypsum or both with the bednar. And over 4 years, the first year we got significant response to the bednar and an additional response to gypsum plus the bednar, but no response to the compost. Years 2 and 3, there were no significant yield responses. Last year, the 4th year, we got significant responses only to the bednar treatments, but not to either of the gypsum or compost additives. Second case study is a different sand. This is deep reddish brown sand at Wotherngara, where we were looking at stacking amendments to see whether we get additive benefits. And I'm just going to focus on a couple of the treatments from that experiment, the control versus spader and the compost treatments. So this is year 5 results. We got a very significant response to the compost, also a response to the spader, but there's no additive effect of the compost and the spader together. So With that, I'm going to now hand over to Stephen to talk about some hard setting or compaction issues related to sandy soils.
Thanks, Richard. Yes, so I'll just be presenting very briefly on some of my PhD research today. So many sandy cropping soils, we see that root growth is physically restricted by high-strength soil layers, and the shallow root growth in these soils then significantly reduces the pool of water and nutrient resources available to plants, reducing yield potential and increasing vulnerability to drought stress. We've seen that strategic deep tillage used to loosen hard layers produce some really good yield responses in many of these soils, but there's also variability we see between sites and seasons that hasn't fully been explained, and that's sort of where some of my research fits in. So I'm looking at both soil and environmental factors that may contribute to some of this variability in how crops have been responding to deep ripping in sandy soils. So we know that roots struggle to grow through compacted soils and that these soils become hard due to surface traffic compressing soil, reducing pore spaces in between the sand grains. And then it subsequently increases the resistance faced by growing root tips trying to push through that soil. But we also know that high soil strength may reflect other mechanisms. So these might be cementation or hard setting processes independent to traffic-induced compaction. So some hard setting soils become impenetrably hard as they dry down due to the cohesion between particles that then collapses again when the soil is re-wet. And so the cause of the high strength problem may have implications for how well or for how long the soil responds to deep tillage. This can be further complicated by interactions with seasonal conditions. So consider this example of a compacted sandy trial site in the mid-north of SA, where roots were being physically constrained from about 20 centimetres. And in the first season following deep ripping treatments at this site, when responses tend to be the strongest, is when we saw no yield gains last year. And if we look at the data, it wasn't an issue to do with the ripping. This graph here shows the cone penetrometer data down the soil profile, and you see that the soil strength is being massively reduced from the red line in the control plots down to the blue line in where it's been deep ripped. And the root growth responded to this as you'd expect. So these heat maps show the root distribution, with roots growing much deeper and denser in our ripped plots compared to the control, where they're really being limited from about 20 centimetres. So then, did these deeper roots actually increase the uptake of deeper water at this site? So this graph shows the soil water content going down the profile for both control in red and ripped plots in blue. And here, early on in the season, you see the same distribution in water down that soil profile. But by about 70 days after sowing, we see here that deeper roots in our ripped plots have certainly increased the uptake of deep water relative to the control. And you'd think that you'd be on track for a good yield response from this. However, in a dry season and with no recharge of this deeper water, we saw bigger plants reaching anthesis in totally dry soil where that blue lines hit the wilting point, meaning that there was no water for good grain set or grain fill going into spring, and so subsequently there was no yield benefit from the ripping. So ultimately it was the timing of the water availability rather than the soil not responding well to ripping that was the problem at this site in this particular season. So variability in response to ripping may be due to both soil and environmental differences between sites and seasons. On the soil side, my research is exploring how hard setting mechanisms may be contributing to how roots are restricted under certain moisture conditions, and more work is certainly needed to explore how deep tillage might be disrupting this process. On the environmental side, it's important to know and understand not only the amount of stored water made accessible by deep ripping, but also the rainfall that's required to recharge this resource, since it's access to this additional soil water during critical stages of plant growth that ripping responses are going to be dependent on. And that's me. I'll pass over to Tanya.
Thank you, Stephen. The challenge I am going to addressing here is the stability of soil organic carbon under different amendments in sandy soil. Okay. We know many of the poorer properties of sandy soil is mostly due to the less organic carbon content, and the general approach to improve the sandy soil health is by adding organic inputs like compost, which can add external carbon input to soil. But in sandy soil, much of this carbon is decomposed quickly and lost. On the other hand, inorganic inputs that can help to protect this added carbon. So my work explores applying these amendments separately or individually or in combination affects the composition of soil organic carbon in sandy soil over time and under different application strategy. Okay, so for one of my experiment, I collected soil samples from a pot trial that was for 73 days, and all the amendments were applied in pelletized form. So after collecting the soil samples, I separated the amendments by sieving, and I extracted and analyzed the amendments using the same procedure as I used for the amended soil. And interestingly, in case of compost, I found that most of the compost fillers remained physically intact throughout the experiment. And the result was quite interesting. I found that total organic carbon compounds concentration was about 6.8 times higher in the compost pellet than the amended soil. That means over a short period of experimental time, these compost fillers act as microbial hotspot with higher microbial activity on the fillet surface than its surrounding amended soil. Then to explore the mechanism of these findings, I set up another follow-up pot trials, extending the experimental period for 6 months. This time, I found a significant increase in carbon compound concentration compared to 3 months. So here it is for the sugar compounds. I also found this increased concentration for other compounds as well. And so this result indicate that the compost fillet break down slowly and they gradually increase the microbial processing and transformation in amended soil. And also we can suggest from this result that compost-based research should be set up for longer time period, although we think 6 months is not still enough time to see the real effect. In my last experiment, I applied combination of compost and hydrotalcite, which is an anionic clay with specific unique surface chemistry. So I observed from the combination of amendments that compost increased the compounds and added more new compounds to the soil, and hydrotalcite protect these compounds by holding them on the surface by physically protection by absorption and physical encapsulation without added any external carbon. So the combination of the compost and hydrotalcite can help to retain the carbon compounds in the soil for longer time periods. And we also found the greater cumulative biomass production over time for the co-application of compost and hydrotalcite than their individual use. Another findings from this experiment was we found insignificant soil organic carbon distribution between two layers of soil profile, that was 0 to 10 centimeter and 11 to 20 centimeter. The variation was less than 5%. So if I summarize the result from experiments and can summarize as the practical recommendation, that pelletized amendments can be more beneficial for long-term carbon soil health in terms of carbon stability, because they can reduce the quick loss of carbon from the soil. And if they applied at the specific depth with seed, not rather than using on the surface. And also compost benefits are time dependent. And finally, compost and hydrotalcite combination can be a potential strategy for long-term carbon stability and nutrient retention. But these points can be strongly proved if we can apply them in a longer term for field trials, considering the cost and the other parameters. Thank you.
Thank you, Tania. I'm going to spend a few minutes summarising with how we can unlock the potential of sands by mitigating low reactive surface area with 4 key questions. First is, what limitations need amending? Good diagnosis is critical here. Are we dealing with subsoil compaction or cementation or subsoil pH, or is it water repellents, or is it nutrients? And often it's more than one of those simultaneously limiting productivity. We haven't talked much about nutrition today, but that is the subject of many other studies. GRDC has been very active in that space, funding work on nutrient limitations in sands. Second question, what amendments are available? Subsoil clay may be, but often it's not. And it's for those situations where it's not available that we have looked at novel products, whether it's from mineral processing, from crushed rocks, or from clay, novel clays. How effective are they? From our results, there's a mixed bag, really. But I think for each of these potential products, we shouldn't rule them out because we haven't yet tested them over a wide range of rates and a diverse range of soil types to really work out where there might be an optimum fit of these novel products for particular soils and particular soil constraints. And of course, the final question is about economics. How cost effective are they to use? We know that subsoil clay can be, but there is quite a long payback period, could be 7+ years before you turn a profit from subsoil clay amendments. And most of these novel products that we're looking at haven't been fully evaluated for benefit cost analysis, but we do have a Soil CRC project supported by Future Drought Fund with Professor Frank Agbola at University of Newcastle, together with Professor Keith Pembleton at University of Southern Queensland, who together are going to help us do a more exhaustive analysis of the benefit cost analysis of these types of amendments. So I'll wrap up there. We have some time for questions, and I'll hand back to Felicity to handle questions.
Thank you, Richard. Yeah, now's the opportunity to ask some questions, and I can see that there are a few questions in the Q&A box. One of the questions has been answered already, Richard, in your presentation, so that's fantastic.
I could elaborate on that.
Would you like to? Yes?
Yes, look, the mineralogy in Southwest Australia, and I think probably South Australia, most of the subsoil clay is kalinitic. but there are some places where illite clays are also dug up. So that's really and kaolinite is not a high activity clay, but it is a clay, and it certainly can be beneficial at the right rates and with appropriate incorporation. And of course, if you can increase the yield potential by 50%, you do need to re-examine what would be optimal nutrition to capitalize on that high yield potential.
Thanks, Richard. Thanks, Carmel, for your next question. Was there a trial where the focus was on building soil biological processes as the primary addition or action, for example, testing the productivity of cash crops after varied periods of resting paddocks with diverse plant species in cover crops with organic nutrients, for example, worm juice added to support the biological population function expansion? Now, Richard, I'm not sure if these trials did that, but we do have another project called the Plant Diversity Project, and I can copy and paste the link to that project. But yes, Richard, do you have a comment about that?
Yes. We have last year done some soil sampling using the Predictor B test of SARDI to look at soil biology and found some very promising differences in our treatments. The compost treatments have shown up in terms of a difference in soil biological activity. So we're going to follow that up because it looks promising. Secondly, one of our trials on a deep sand does include some mixed species cropping and rotational effects. And at that trial last year, we did get quite a significant yield increase in our canola crop that followed a mixture of vetch and lupin in the previous year. And we attribute that to improved nitrogen availability. So yeah, that's probably the extent of the work that we've been doing so far on the biological aspects.
Thanks, Richard. And Gabby from West Midlands Group, they've got a bit of work ahead of them with a 5-year project looking with sandy soils in the West Midlands region. Is there any really new or novel things people or groups are doing that we could follow? I'm not sure, Richard and Amanda, if you've got a comment about anything new or novel that's happening in South Australia that the Western Australians might be able to latch on to.
We probably don't have anything novel. People are looking at addressing the constraints, so it's the usuals. Biochar has been tested by a number of landscape boards about looking at those, and as Richard was saying, it's the feedstock and the rates that are really important in that. And there are some FDF long-term trial projects that are looking at more the diverse crop examples, I suppose, but no novel amendments from above and beyond what Richard's already got going at West Midlands.
Amanda, any comments?
Just as biochar is a generic term for a wide diversity of products, so too is compost. So there are many types of compost, some of which are nutrient rich and break down quite quickly. Others are quite recalcitrant, and some compost products have clay addition to them. So I think it's a matter of horses for courses and trying to fit the right product for the situation. One of the interesting things, Gabby, about the Wathingarra trial that your West Midlands group's been looking after is that it's a relatively high rainfall site and high productivity potential site. And that's the one where we've got the response to compost, and it sort of changes my thinking a little bit about where there's a role for compost, from thinking it's probably on the low productivity sites. But maybe the big gains are to be made where there is a high productivity potential, and the compost is just able to improve the water and nutrient supply to the roots to capitalize on that potential. But I suppose the other thing I would say is that Around rural Australia, you often find localised sources of organic matter. It could be around piggeries, poultry sheds, and where that local supply of organic matter is available, within a radius of perhaps 50 kilometres around that, there's real potential to use those resources as soil amendments. Probably not going to be economic to transport them over much longer distances, but we know in Western Australia there's quite a few of those intensive animal industry operations where organic matter could be sourced for sandy soils.
And can I just add, there was another trial too that was done around Coombe, so our lower southeast kind of lower Mallee area there, that also had looked at Great Mark, and that's particularly of interest for potentially potassium. So it was a high source of potassium. So again, if it was a local waste treatment as such in an area, that was well done. And we've got some really good examples, like Richard was saying about chicken manure, pig manure in the areas, as long as they have some kind of composted effect, so that they're not just raw going on. They've been very effective.
Thanks, Amanda. Fantastic. And Bridget from Western Australia, the Regional Soil Coordinator in Western Australia, has really asked a really important question around what are some of the soil characteristics landholders should assess before excavating and spreading on farm clay source to avoid negative consequences, and what analysis, etc., should they ask to the lab to run to ensure they're getting a quality product? Richard, would you like to have a go at that one?
Look, I think the main thing is that it's got to have enough clay in it to warrant the cost of putting it on. If you put on 200 to 300 tonnes of subsoil and it's got 30 to 35% clay in it, you're putting on about 70 to 100 tonnes of actual clay. And that seems to work in the higher rainfall zones. In the lower rainfall zones, slightly lower rates do seem to be sufficient. So the first thing is just to check the clay content of the subsoil. And look, it's likely to be reasonably similar over a region or over a farm. And that simple ribbon test that I illustrated is one way of doing that. You could take the sample to the lab. But that would be the first thing I would do. David Hall did some rough calculations on what would happen if you added a clay that was a bit salty or sodic or high in boron. By the time it's diluted and mixed into the soil, that's probably not going to be a limitation. So it's going to be more around the clay content, but some of the subsoils can contain very high levels of potassium. In fact, you're basically putting on enough potassium fertilizer to keep you going for probably a decade or more in the subsoil, but other subsoils contain no clay. So if you don't understand those differences, you won't know how to adjust a fertilizer program accordingly.
Thanks, Richard. The next question from John is around irrigating somewhat saline water and what difference that might make to which amendments would be most beneficial, with the concern that possibly, or the assumption that possibly, clay amendments may hold salts in the root zone. Yeah, Richard or Amanda have a comment about that?
Look, I could make some comments. If you are irrigating, then presumably you've got the capacity to apply enough excess water to actually leach salts out of the root zone. So I think that would probably be what you'd be thinking about, as you would do with any best practiced irrigation, because almost all irrigation water contains a little bit of salt, and so you've got to have that a little bit of surplus water added to keep those salts leached below the root zone. As Amanda said, the sweet spot for clay addition is 6 to 15%. And when you mix the clay into sands, you'll tend to result in clay in clods or lumps. It's not a uniform powder mixed into the soil. So, yeah, I would think you can probably manage without causing a salt problem, so long as you're adding enough extra water to do that, have a leaching fraction.
Right, thanks, Richard. And one last quick question around how the iron content in sandy soils might affect the hardening or the development of a hard pan layer in the textured soils and sandy soils, and how organic amendments may affect the hardening.
Sounds like a Stephen question.
A Stephen question, yeah.
Yeah, I can jump in on that.
Thanks,
that one
Stephen.
sounds good. I guess, yeah, two parts to that. So the organic amendments in the experiments we've been doing, and certainly some of the other ones people have done in the past, you see a negative correlation there where, yeah, you increase organic matter in hardening soil layers and they don't harden as much as they dry out. So that's certainly an effective way of reducing that hardening. Whether it's always cost effective might be another issue. On the iron front, that is, yeah, an interesting avenue to go down. So there's certainly evidence of iron oxide coatings on sand grains potentially contributing to strength of some sands as they dry down, but how settled that is or how widespread that phenomenon is probably needing more exploration, unless someone like Richard wants to comment on that. But yeah, like that, it can contribute, but it's not the only factor at play.
Thank you, Stephen. Yeah, did you have any other comments, Richard, or we might
I'll leave it there. I think we're out of time anyway.
Yes, we are. Wonderful. Thank you very much. And thank you to our very informative presentations and presenters, to Richard, Amanda, Stephen, and Tania for sharing your knowledge on sandy soils. And thank you all for your interest in attending. Please complete the survey as you exit the webinar. This has been recorded and the resources will be sent to your registered email. And I look forward to seeing you all at the next Soil CRC webinar. So thank you all again and good afternoon.